Yeah, it's kind of small. Did you have the little bio you wanted to do?
Oh, I didn't print it out, but
You didn't print it out?
no.
That's okay. I can, uh, well, or you can do the self-explanation. USF?
Yes,
Biology?
environmental science
Environmental
policy.
science. Okay. Okay, well, welcome. Thank you for attending. Today is the master's thesis defense of Robert Schroeder. Robert came to us after an environmental studies degree at USF and then an internship for a year at USGS. And who was it you worked with there?
Arturo Torres, Patty Metz, Kim Hag, Terry Lee, Amy Swankar. Anybody?
So then after that stint with USGS, Rob came to FIU where he started working actually in the lab, working up at Lila with Eric, who at the time was, you were still working for me here, right? Yeah, up there at Lila. And so he worked there for a year and then he started into the master's program. And as many of you know, he's been kind of integral to all of our work up at Lila. In fact, we worked with Suresh and Alexandra, Diana. So he's been working up at Lila now for a couple of years, and so he's in. So basically, some of the work we've been doing up there, looking at gas exchange, he'll talk about today, is his thesis work.
So first, I'd like to thank everybody for coming today, and hope you enjoy the presentation. And this will be my master's defense entitled Soil Carbon Dioxide, or CO2, and Methane, CH4 Efflux, from an Everglades tree island and ridge landscape. I'm part of the Earth and Environment Department, and I'd also like to thank my committee members, Dr. J and Dr. Oberbauer, and most importantly of all, my major advisor, Dr. Sento. First, I'm going to give you a brief introduction into the Everglades itself and why we're studying what we're studying. Then I'm going to go through the study site and why it's crucial to understanding these gas dynamics in the Everglades ecosystem. Then I'm going to go through each of my hypotheses with methods, results, and some discussion. Next, I'm going to look at some of the implications for all of this, and then finally, I'm going to make some conclusions. So, beginning in the early 1900s, we started digging canals and levees for expansion of agricultural land. The Everglades were thought to be very profitable for farming and later development on the eastern seaboard because of the peat soils that accumulated over thousands of years of development. And by the 1970s, we had about 2,500 kilometers of these canals and levees. Now, this brought about some spatial changes to the Everglades ecosystem itself. We lost about 55% of the total spatial coverage. It allowed for this large development of Everglades agricultural here south of Lake Okeechobee, and this large development along the east coast that is the Tri-County area that we all know and love and live in. Now, the canals and levees divided it into three main sections, conservation areas, 1, 2, 3, and Everglades National Park down here at the southern end. This is all federally protected land here that remains. However, the canals and levees that crisscross it also introduce new things to the ecosystem itself, namely hydrologic changes. These changes have reduced the normal uniform water depth into these ponds, in essence, that have a more uniform water level compared to the more uniform flow and slope to the land. This creates these really dry sections during the dry season here, and these really wet sections during the dry season when water levels fluctuate in the Everglades due to precipitation patterns. What it also has done is reduce the flow itself. We now, instead of water flowing from north to south into Florida Bay and all the estuaries down here, it now ends up out in the Caloosahatchee and the St. Lucie waterways, or along the east coast here through the main development areas. What has all this reduction in flow done to the system itself, and subsequently also the loss in water levels? We've degraded the normally homogeneous ecosystem of the ridge and slough landscape. The ridge and slough landscape was dominant in the Everglades and developed parallel to flow with these deeper sections or sloughs, and that remained wet throughout most of the year, versus the drier ridges, which had slight elevation rises compared to the slough and only experienced partial flooding throughout the year. The reduction in flow and also water levels has created a more homogeneous landscape here. We have also part of this ridge and slough landscape are tree islands. Tree islands were home to many mammals in the Everglades itself. Because the Everglades is a very wet ecosystem, tree islands were relatively topographical highs, about 90 centimeters greater than the sloughs itself. And this kept these areas drier throughout the year and provided homes for these mammalian species and wading birds and migratory birds themselves. Since the 1940s, there's been about a 67% reduction in overall total tree island coverage. What mainly has happened is we've lost a lot of the spacing between these tree islands, making it more difficult for the mammalian species to travel to and from tree island to seek refuge. Now, SERP was enacted, or the Comprehensive Everglades Restoration Plan was enacted in early 2000s to help restore the Everglades and in quality, quantity, and timing of the water in the Everglades itself. One of the things not included in the plan itself was the inclusion of tree islands, mainly due to many lack of understandings on how they develop, maintain themselves, and built initially. So what this presented an opportunity for researchers both at South Florida Water Management District, Fish and Wildlife Service, and the Army Corps of Engineers to do is develop a living landscape, which is the Loxahatchee Impoundment Landscape Assessment, LILA, right here on the eastern side of Loxahatchee National Wildlife Refuge and Boynton Beach. LILA is constructed out of two main catchments itself that were originally farmed up until the 1970s, and what they did is they excavated the peat that was in these catchments and built tree islands here, ridges here, and where they excavated the soil, they created sloughs. So we have the three main habitats or ecotones associated with the Everglades. They built 4 of them total, and one of the things we wanted to get was quality, quantity, and timing. So we created, and the other thing we've done in the Everglades is reduce the flow. So what we can do here at LILA is actually collect water here in a canal on this side, the eastern side, pump it into a canal here on the western side, and actually flow through the cells itself. There's stage recorders at the inflow and the outflow, so we can get real-time stage across the entire cell. And we also have flow monitors based on how much opening we have in the gates. So we can correlate what is happening biologically in these cells to stage flow. Now, just a cross-section of what each macrocosm looks like. We have the general relative elevation here in meters, and my 3 sites that I studied are head high on the top of the center portion of the tree island, which is about 5 meters in elevation, the head low, which is halfway down the slope of that tree island, and that's about 20 to 30 centimeters lower than the head high. And then finally we have the lowest elevation I studied, which is the middle ridge, the next lowest elevation. Now, LILA is maintained by an operational hydrograph. This was constructed over from 30 years of stage and rainfall data in a northern Everglades setting to develop where we want the hydrograph or the water levels throughout the entire year. We try to Eric here tries to maintain these water levels as best he can, depending on what happens with the pump. And you can see here where we have our tree islands and ridges elevations here. So I just wanted to introduce all of this stuff to you, the head high, HH, head low, HL, and the middle ridge is MR. Now in 2006 was the planting of these tree islands itself. So these are relatively young. We not only constructed them with excavation equipment, we planted the trees themselves in a grid pattern with multiple different species and multiple different densities of planting. By 2010, we were starting to see a decent canopy develop, and on one tree island that we're studying, M1, which is a peat core tree island, and I'll get into that in a moment, the canopy developed a lot faster. So what does all of this mean? Well, we're trying to define how because we can control the water levels not only at Lila, but also in the Everglades through all the man-made structures, we're trying to understand how tree islands and ridges develop and maintain themselves based on water levels. Oops. Okay. We have a decomposition curve here, which just indicates how much organic matter is decomposing and being expelled as gaseous emissions. And this is maintained by relative water level as well, along with other things like organic matter quality, extracellular enzyme activity, which just basically tells us how much microbial activity is in the soil. The other curve of this is the production curve, which is how much the biomass or the plants are producing on these tree islands and ridges. What we're trying to define is this area here where production can build tree islands, and also where the equilibrium points are on compared to relative water depth, where decomposition equals production. This will enable us to better control water levels not only at Lila, but in the Everglades proper itself, to maintain or even rebuild tree islands that are critical for Everglades restoration. And on this study, I'm mostly focusing on the decomposition aspect. Now, this is an entire hydrograph of the study period that I did for the 2 years. And what I want to highlight here is what we're seeing here is the rainfall picks up in April through September, but the water levels lag behind by a couple months, mainly because it takes time to accumulate all this water and actually build the water levels. And so what we're going to define here as, because I'm looking at water levels itself, is wet condition versus dry condition. And wet condition here is what we're going to define as September through January, when the water levels are at their highest levels throughout the year. And then we're going to define the dry condition, which is April through July, which is when the water levels are at their lowest. And we can also see here that in dry condition 2011, we actually had one of the lowest periods of record on stage. I was out there, all the cells dried out, and you could walk all the way across the cells. It was quite interesting. Now, what I've also defined here is where, in relation to my three study plots, are on this hydrograph. We have the head high, which, as you can see, did not flood throughout the year. We also have the head low, which has considerable amounts of flooding, and then also the middle ridge, which is flooded predominantly throughout the year. And highlighted in these vertical gray bars are my study times. So what do I want to do here through this research? Is I want to determine if water levels are a main driver of sea loss from the Everglades tree island and ridge landscape. Also estimate how much carbon loss we are losing from these Everglades tree islands and ridges. Determine whether or not nutrient or organic matter quality is limiting respiration from these tree islands, because we do know that the Everglades are nutrient limited. They're phosphorus limited. They're oligotrophic. Does this inhibit respiration from these tree islands, or is it more on organic matter quality, which means that how much carbon can they easily extract or pull out of the soil? And finally, determine what kind of microbial activity is going on in the soil. Extracellular enzyme activity is an excellent way to do that. And see if any nutrient or organic matter quality limitation plays into the way we're seeing these activities change, both seasonally with water conditions and with changes in those nutrients. So I'm going to start with my first two hypotheses because they kind of tie in hand to hand together. First is going to look at CO2, my carbon dioxide respiration on tree islands, and I'm hypothesizing that it's negatively influenced by water level itself. I'm also suggesting that organic matter decomposition by methanogenic pathways or methane production is negatively influenced by water level. So first, I'm going to look at the in situ or in the field measurements of my CO2 efflux. The way we decided to go about doing this is with the LA8100 from Lycor. This is an infrared gas analyzer, or an ERGA, and it tells you the concentration of CO2 in one of these chambers when the arm comes around and closes, and as Eric and I have affectionately called them, R2D2s from Star Wars. Only nerd? Okay. And what the ERGA tells you is the concentration of gas over a certain period of time that you have the chamber closed. And based on the linear rise in CO2 efflux, or CO2 in the chamber, it calculates an efflux rate. And it corrects for pressure and a host of different other things going on in the chamber. Now, all of the collars that I sampled, which are a set diameter, are all XYZ located on the tree island, which is a very big benefit to Lila itself. I can go out, get a very accurate GPS coordinate to calculate elevations, calculate water levels at each one of these points. And because of knowing the XYZ and knowing the water table, or the water levels in the cells, I can calculate a relative water depth, which is just basically my stage of the cell subtracting the soil elevation. And I'll get into that in a moment. So I took 4 different samples throughout the year, measuring 4 major points of the operational hydrograph, which is low water levels, rising water levels, high water levels, and falling water levels, to see if there's any changes with the water condition throughout the year. So just another reiteration of my head highs, or HHs. They were approximately 5 meters in elevation, and all of the head high plots had the same elevation, so there was no variation in these plots. They also received no flooding throughout the 1-year in situ study. My head low plots had almost 20 centimeters of difference between the highest one and the lowest one, and they received between 144 and 289 days of flooding throughout the year. And I'll get into that more so in a moment. And finally, the middle ridges, the EMRs, they had approximately 4.45 meters of elevation, and also, again, about another 10 centimeter difference in elevation. But they had between 292 and 349 days of flooding, so they are flooded the good predominant amount of the year. Now, how does all that tie in together? First, on my y-axis, I have my efflux rate in micromoles of CO2 per meter squared per second. On the x-axis, I have my relative water depth. Negative values of relative water depth indicates water level below the soil surface, where positive values indicate water above the soil surface. And what we see here is each one of these plots is negatively influenced by relative water depth for CO2 efflux. Now, the scales of efflux are different for each one, so do note that. And what I'd like to point out is, especially on the head highs, which did not receive any flooding, we're getting a very large difference in rates of efflux. Now, these 2 tree islands were, one was a peat core and one was a limestone core. In the northern Everglades, tree islands are peat dominated. They're called battery tree islands. Whereas in the southern Everglades, they're more limestone oriented because of the way they've developed behind limestone outcroppings. And this may be an indication that we're seeing a difference in how they behave when water levels are much lower than the soil surface. And so what can we do with this information? Well, we can combine it. This is just for my tree island efflux only. No ridges were included. What we can do is combine both of these into a single model. I defined each season's or each measurement times efflux by a different color on this, so you can see when water levels are high or when water levels are low, the efflux is generally higher than when they're at their highest in September. And again, non-flooded is negative values, positive values are flooded. What we see is that we do get a very significant curve here when all values are included. What we also get is another equally significant curve, but with a slightly higher slope when we don't include that M2 lime core tree island efflux, which is down here, bottom of the line. So we may be seeing a difference in how they behave when water levels are lower than 60 centimeters below the soil surface. So what can we do with all of this information? We know the stage throughout the entire study period, as I previously showed you, throughout the year. And I can take those equations that I generated by each plot and calculate an estimate of carbon loss as grams carbon per meter squared per second. And what we're seeing again is that the head highs, which received no flooding, had higher rates of sea emissions than the head lows, which had longer periods of flooding. Then also we have the middle ridges, which have even less efflux from the soil surface, and they receive the most amount of flooding throughout the entire study period. Now, one of the things to note is we did have high N values. However, we did also have some lower R square values. And there's several different plays going on with that. Biomass is another large driver of sea efflux from the soil surface itself. And I'll get more into that in a moment. But we do see that values from Lila are in line with other values from literature. Hirano and Huainan are all from Malaysian peat swamp forest, and Meling is from another Indonesian swamp forest as well. So biomass has been shown in several different studies to influence the rates of CO2 efflux from the soil surface. And another one of the main benefits of Lila is that we can correlate between different lab groups and within lab groups to come to a more integrated understanding of the system itself. Part of another graduate student in Dr. Sento's lab is looking more at the litter fall of soil and how that impacts the soil development itself. And I can take that same data and plot it against my efflux and see how greater amounts of litter fall are influencing my sea efflux. So we're seeing with more litter fall, which also produces more carbon onto the soil surface, we're getting higher rates of sea efflux. So with larger carbon inputs, we're also seeing more efflux. And again, we can see the M1 is has higher rates of litter fall and also has higher rates of efflux. The other thing is Dr. Ross's group calculates biomass. They just moved to annual calculations of biomass, but they also had calculations 2 times a year going back to when I was doing my research out there. And what we also find is that biomass is influencing the rates of sea efflux. Now, this has been shown by Krauss et al. Before, and we're seeing very significant rates of increase with both biomass and litter fall into CO2 efflux. And while we're not seeing very strong linear correlations with water levels, we're also, as far as our squares go, we are seeing some stronger correlations with biomass here. Now, one of the other ways I calculated CO2 efflux and methane efflux was in a core incubation study. I took 20 centimeters of soil in triplicate from each of my plots and used the lichor itself to calculate CO2 efflux. I also pulled gas samples from the cap here and was able to analyze them for methane efflux. I drew the water levels down after a period of incubation and equalization in each water level, and I was also at the same time measured for oxidation reduction potential. Now, one of the things that water levels do when high is reduce the availability of oxygen to get to the soil, and oxygen is the most powerful form of electron acceptor to produce energy for the microbial community. Without oxygen, they go through a series of different electron acceptors, and you can measure where on where they are on the millivolt scale of redox potential. You can kind of guesstimate as to where they are on their um, electron acceptors. Finally reaching negative 200 millivolts, which is considered the threshold for methane production. However, these are modified systems. I disturb the soil, and so it's not going to behave the same way as what would happen in the environment itself. But it allows me to correlate a lot of different things into one thing, as well as minimize one of the major variables, as I just showed you, which is the tree influence of root respiration. Now, first is my carbon dioxide efflux, which was significantly influenced by relative water depth or water level in the core tubes. It was also significantly influenced by the elevation, which is just another iteration of the highs having more production than the lows than the middle ridges. What it also allowed me to do is calculate how much we're accounting for in this core study from the in situ efflux. What I found is that we're finding between 16 and 34% of the tree island respiration coming solely from the core tubes. Whereas on the middle ridge, which don't have trees on them, I'm accounting for between 29 and 54% of the total respiration. Theoretically, we could use this as the microbial contribution to CO2 efflux from Lila tree islands and ridges, but further investigation is warranted into something of such a point. Now, the next thing I did was look at methane efflux. What I did find is that water level is not a significant influence on methane efflux from this landscape. This is something DeBusk and Reddy have also shown from WCA2 soils, Water Conservation Area 2 soils. What I did find is that where the soil comes from, head high, head low, middle ridge, is a significant driver of how much methane is produced from these soils. Generally speaking, the ridges had greater production from the tree islands. They also are have the longest period of flooding, which means they also have a greater ability to develop methanogenic bacteria. Finally, the redox potential. Theoretically, a lot of papers like to use redox as a potential way to delve into the carbon cycle, because we can understand, based on the redox scale, where we are on CO2 production versus methane production and how that all influences it. However, I did not find any direct correlation between methane production and redox potential from these soils. What I did find is that water level is a significant driver of the redox potential, which, when water levels are high, we're going to have low diffusibility of oxygen in the system, and oxygen is going to become limited. So that's understandable. Also, elevation was a big play, and also their interaction. So what can we conclude from this? What we conclude is that soil CO2 efflux and respiration on tree islands and ridges is significantly influenced by water levels in these systems. However, the methanogenic pathway of organic matter decomposition is not significantly influenced by water levels. We can also say that between 16 and 54% of the in the field measurements of tree island and ridge CO2 efflux is accounted for in the core study, and that redox potential was also significantly influenced by water, but did not show a significant influence on CH4 or methane efflux. Now moving on, CO2 and what I proposed here was that CO2 and CH4 production from the tree island and ridge landscape between the wet and dry condition soils varies with organic matter and labile phosphorus additions. What this in essence means is that we know that the Everglades are phosphorus limited, including the tree islands themselves, at least at Lila. As I'll show you in a second. Some of the tree islands in the Everglades themselves are not phosphorus limited. However, we do know that less recalcitrant forms or less usable forms of carbon in the soil itself that are build up over time due to their ability to not be decomposed. The tree islands themselves have higher elevations in the Everglades than the ridges, so that in essence means that maybe they have a strong recalcitrance of organic matter, and see how that plays out. So what I did is I took wet and dry conditioned soil cores and incubated them in incubation vials. I measured them for 5 days, and I amended them with various forms of with a labile carbon form of glucose, as well as salt of phosphorus, and I combined them as well. Now, the control had a carbon to phosphorus ratio of 675 to 1, which means for every molecule of phosphorus, we're going to see 675 carbons first. Now, the Redfield ratio that states the ideal amount of carbon to phosphorus is 106 carbons to 1 phosphorus. So this indicates that we're severely carbon or phosphorus limited on these Everglades tree islands and ridges. However, when I amended them with phosphorus, I reduced that limitation down to 116 to 1. While not completely down to the Redfield ratio, I got close and substantially reduced the phosphorus limitation. However, when I added the glucose or the carbon source to these soils, I only slightly increased the phosphorus limitation. And then again, with the carbon and phosphorus addition, only slightly increased it over the solely phosphorus enrichment. So I'm not only producing a new source of labile carbon that can easily be uptaken, I'm also substantially reducing the phosphorus limitation. So what did we learn from this study is that we're seeing a significant response to well, first let me explain. On the y-axis, we have my CO2 production rate. On the x-axis, I have my dry and wet conditioned soils, or which condition they were from. And then my blue is control, green is my glucose addition, my gold is my glucose and phosphorus addition, and my purple is my phosphorus addition only. So what we're seeing is that we're getting substantial, significant increases in CO2 production from, especially during the dry season, with any form of glucose enrichment. Now the only plot, which is my head low here, to actually show a phosphorus treatment effect was the head low itself. No, the head highs nor the middle ridges showed a phosphorus enrichment treatment effect. But the glucose and phosphorus enrichment in the middle, we can see is, from the head high and the head low was significant in both wet and dry conditions, and only the middle ridge showed the effect in the dry conditioned soils. So this is an indication that we are carbon limited from, or carbon lability limited of respiration from these soils, and not phosphorus limited, except in this head low plot. And they're only slightly, they're significant, but they're only slightly different. Now, due to Suresh's work out here at Lila, he was amending the soils to see, with nitrogen and phosphorus, to see what happens to tree uptake, and where they store and how they use these nutrients. I was able to sample the soil itself for CO2 efflux at the same time and get an idea of what a control versus a phosphorus treatment is doing to the soil efflux rate. And what I find is that there is no significant difference between the two. And this is just further indication that phosphorus is not limiting respiration from these soils. And additionally, I also analyzed them for methane production, and we found the same treatment effect here, which glucose and phosphorus enrichments definitely do, or significantly do increase respiration, more so in the dry season than the wet, from these soils. Excuse me 1 second. So what can we conclude from this? Well, we learned that the carbon dioxide and methane efflux from these soils is carbon limited for sure, significantly, and that only the only the head low also only showed a labile P limitation as well. And finally, the extracellular enzyme activity. With these changes in availability of phosphorus and carbon, are we going to see a response from the microbial community itself as well? And the way we can do that is measuring extracellular enzyme activity, which just measures the activity of enzymes in the soil that are used or produced by the microbial community. Higher rates of enzyme activity, as I'm about to show you, are indicative of more microbial activity in the soil, begetting more decomposition rates. First is glucosidase. Now, this is used to break carbon-carbon bonds in the soil matrix itself to pull out glucose from the soil matrices. And what we find is that there's only a significant increase in glucosidase activity in dry conditions with glucose and phosphorus enrichments, which just shows that these systems, when that phosphorus is not limiting their use of this enzyme, it's more due to the lack of labile carbon source, which is something. Labile carbon is something that has been shown to be limiting respiration in Everglades soils like Amador and Jones and Dubusque and Reddy. Now, what does this glucosidase activity show us? Well, it's been shown by other sources that glucosidase activity correlates to CO2 production. And we see this trend here as well, both in the wet and the dry conditions, dry being blue. And we can correlate rapidly our glucosidase activity to CO2 production and even CO2 efflux if we core and measure CO2 efflux at the same time. And the enzyme activity is thought to be a good soil quality indicator. And the microbial community is generally the first to respond to changes in the soil and any biological, and first to respond to any new stimuli, when we could rapidly determine what is happening with changes in water levels or soil phosphorus or any such thing. Additionally, phosphatase, that's used to break carbon-phosphorus bonds. We know that phosphorus is limiting in the Everglades, and so we should see relatively high rates of phosphatase activity trying to pull phosphorus out of these soils to enhance the microbial community. And what we see here is, especially during the dry conditioned soils, we see inhibition of the phosphatase activity when any form of glucose or any form of phosphorus is added to the soil, which just indicates that when a labile phosphorus form is added, we're going to drop phosphatase activity because the microbial community does not have designated resources to its activity. While not significant, what we see when we add just the glucose itself is a slight increase in the phosphatase activity. That's just because labile phosphorus now becomes an even more limiting when we reduce the labile carbon availability. So from this, what we can conclude is that glucosidase does show from head-low soils that labile carbon and phosphorus enrichments do increase the activity, while the head-high and the middle ridge soils do not have a difference. Additionally, while wet and dry conditions, they do significantly correlate to CO2 efflux with glucosidase activity. And finally, the phosphatase activity was significantly inhibited with any labile P additions. So we can also use phosphatase as an additional source of soil quality indicator if a labile phosphorus source were to impact these soils. So what does all this do tie in together? Well, we can use the models generated here and test them on Everglades tree islands out in the actual Everglades. And we can use them to determine what changes in water levels due to the rates of CO2 and methane efflux. And just for the sake of testing this, I changed the stage throughout the study period by 0.03 meters throughout the entire study period and recalculated my estimates of sea efflux. With a rise of the 3 centimeters, I got an additional decrease in sea loss from these soils between 74 and 160 grams carbon per meter squared per year. Conversely, if I dropped or lowered the water levels the same amount throughout the year, we saw an increase in the rates of sea loss by the same amount. So this just indicates that we can minimize how much sea loss or carbon loss from these Everglades tree islands and ridges. We can minimize it by managing our water levels properly. Now, phosphorus limitation has not been shown to be inhibiting CO2 production, but increases in labile C concentration have shown that there could be a potential to have higher rates of CO2 production. But long term, we need to continue to monitor these systems because the Lila tree islands are young, they were constructed, their stuff is still going on, and they're developing. So we need to understand long term how CO2 efflux and the enzyme activity changes in these soils as the tree islands develop. We can use all of that information combined to better help the restoration in the Everglades of these tree islands and ridges, and know how they develop and can best manage according to what we learned from Lila. So, just in conclusion, for my hypotheses itself, we did find that CO2 efflux is significantly negatively influenced by water levels from all study plots and cores. We also found that methane is not significantly influenced by water levels itself, that both CO2 and methane production increases with labile carbon and phosphorus combination enrichments. And then finally, glucosidase was not enhanced by labile carbon or phosphorus enrichments, and phosphatase was significantly inhibited when any form of labile P was added. So I'd like to thank you all for coming today. I'd like to take a few moments to acknowledge the people. Dr. Sinto, without you, I would not have been able to have done this research. You've been integral in helping me along. Dr. Jay and Dr. Oberbauer, thank you for being on my committee, and your knowledge has been great. Eric, Tom Dreschel, and Fred Sklar at the districts. Without you guys, we wouldn't have the Lila contract, and you guys have been awesome. Alexandra, thank you for your help in the beginning, especially moving the LICOR, and more so with the writing and understanding statistics. Diana, without your help in the lab, I don't know what I would have done. Ryan, he was used to work up at Lila. He helped me a lot out in the field. I'd like to thank my family that's here, my mom and dad, as well as my brother who's up at Gainesville, all my friends here, and then my former employers at the USGS. So thank you all. I'll take any questions.
So just as a matter of course, what we're going to do now is you guys can ask questions. I think the committee will reserve questions unless you want to open a discussion. And then everybody will be asked to leave, and then we'll grill Rob until he's frantic. and then we'll see what happens. So if anybody has questions.
You go back to the carbon flux from the head. That one, you measured the LICOR.
This one?
The beginning. No. I wonder because.
This one? Or this one?
Whatever would be good. Go to the next one, I think. I wonder because, I don't know, I didn't figure out here, but I heard that. So you measure the carbon, so you find the carbon dioxide more in the highest resolution, like the head?
More efflux?
Yeah.
No. Here in the middle ridge, we actually got some really high rates of efflux. But more so, it seems to kind of level out around 10, which is generally some of the highest rates in literature that I found.
It's in the middle, right?
But that was, when they dried out, those plots experience real long periods of flooding and then they're dry for a short. So they have potential to have built up lots of labile carbon and it just goes up. What we do find here, especially on the M1 head high versus M2 head high, that efflux at the lowest water levels is much higher on the M1 head high, which, as you know, is the peak core tree island.
Because I'm wondering because the head is kind of your experiment period is never flooded, so it's basically a dry, right? But in the middle of some
While it doesn't necessarily get flooded per se, the peat is really, as I'm sure you're aware, during the wet season, it's like real soft and stuff out there. So we are getting some close to saturated conditions on the tree islands, and you can see even still the negative influence that water levels do have.
Right. My question is, I wonder because this microbial activity is, I think some of the research in estuaries in some part they talk about the frost of this water sometimes go on and off can have created more efflux than the one that is most of the time dry or wet.
Right, and I delved more into that in my thesis itself. A lot of times, especially on the peat tree islands, you can build up lots of CO2 and methane when water levels are high because it's not going to diffuse out, and that could potentially be, as water levels fall, just diffuse out and efflux out, and that's something I delved some into my thesis itself. I didn't really look at I didn't look at what was dissolved in the water column itself, both on the tree island and in the sloughs. So that's something for more research.
So you find any difference between the islands, like
Yeah, especially on the head highs, the head lows were only slightly different, but the ridges did behave more or less different. Dr.
Yeah.
Ross? Oh.
So you're measuring efflux in a chamber, and in some cases there's no water on the surface, and then in some cases there's water on the surface.
Mhm.
So when there's no water on the surface, you have two. You're just exchanging from the soil into the atmosphere, and the other one you have to go through the water column.
Right.
So how do you deal with that when you put all these things together?
We developed these, Dr. Sinto was more so, we developed these floating chambers itself. Now, we did not get into the pH differences in the water column because that can drive CO2 efflux, the diurnal cycle of pH due to photosynthesis and especially with the periphyton here in the water column. We're just counting for the diffusive rate out of the water column. We're not, that's for more research to get into what's actually driving, when they're flooded, the rates of efflux that I measured.
But was there a time when your collars were out there when the soil was underwater, but you still had a collar and you were still doing measurements?
Yeah.
So there's some difference between when there's just open airspace and that,
Correct.
and you don't adjust your data for that?
I have longer columns that I was able to put into the soil in the same spots and let those set up before I actually measured on these flooded plots. All I'm accounting for when the quote unquote flooded is what the diffusive rate out of the water column is. It's not what is actually coming out of the soil itself. That's something we sort of looked into but didn't. Dr. Sinto did actually do some experimentation with out on the ridge with some of that and the slough. It's just when you get into little bits of flooding, it's really hard to set up an inverted trap and actually trap the gas coming out of the soil solely. So we didn't get too much into what's coming directly out of the soil.
Could you go to the slide that you presented
water you look at the treatment.
Because yeah, this looks weird. Because the efflux, the phosphorus treatment didn't have any effect. It's pretty much because I measure
We-
this
Right.
couple-
Now,
couple
the
of
other
months
thing with
ago.
this that I didn't get into is when I was doing this was when it was severely severe drought that summer. So there may be other factors going on here, like when you added the nutrient mix of water, it just poured out the bottom of your your PVC into the ground, and it really didn't enhance the phosphorus of the
Right.
soil. It just went straight down.
Because I measure the photosynthesis and stomatic conductivity in a couple of months. I have my graphs that I analyzed this week, and it really shows that the one that I treated with phosphorus could have also high conductivity in terms of the stomata and also have a high photosynthesis. So it's kind of, I'm thinking of now interpreting the other way, like, okay, microbial activity is going, crazy, and then the plant's getting more nutrients, so it might be so that we can talk about this, but Mike, Dr. Sinterville wants to, or Mike Ross wants to do more work on these things, maybe.
Yeah. That would definitely be something to really look into. Because especially showing that biomass is
Right.
in influencing my efflux. So
Okay, thanks folks, thanks for coming.